Hey everyone, how's it going? Here to talk about our next topic. We're getting into protein synthesis a little bit. We gave some examples in the last lecture, and again we talked about what do proteins do and what are examples of proteins and why are they so important. Now we're going to get a little bit into the nitty-gritty of exactly how we make proteins. We're not going to get into too many details, but enough so that you can understand what's going on. And so what we're going to do is we're going to talk about transcription, then translation, and then finally the universal genetic code. So let's get started. Obviously, we've been saying over and over again, DNA makes RNA, makes protein. When DNA makes RNA, that's called transcription. And when RNA makes protein, that's called translation. So make sure you understand that and get those straight in your head. Now, one way you can remember these two, DNA and RNA are very similar molecules, and we'll see that here in a second. And so when you transcribe something, you're kind of keeping it in the same language. You're just sort of writing it down. And so when we go DNA to RNA, those two molecules are very similar, so you're just transcribing them. Translate means to take from one language and put it into another language. And you'll see in a second here that proteins are quite a bit different than RNA. First off, you realize, protein's a class of molecule and RNA is a nucleic acid. They're two different classes of molecule. So when we go from DNA to RNA, it's the same type of molecule. So you're just transcribing. But when you go from RNA to protein, it's two different types of molecules. And so it's like going from one language to another language, and to do that you have to translate something. So that's one way to kind of keep track or keep these two straight in your brain. So a gene is transcribed into a piece of RNA, and then that piece of RNA is translated into a protein. And so remember that that's the definition of a gene, a part of a chromosome that gets transcribed. We said a gene is a part of a chromosome that gets turned into protein. Now we're getting into a little bit more detail. How does it get turned into a protein? It first gets transcribed into RNA, and then that RNA gets translated into protein. And so I just said this, so this is a leftover clicker question from before, but what's a gene? It's a piece of a chromosome that codes for a protein. Now, here's a new question. If you were to guess about how much of your DNA is not a gene, that is, how much of your DNA does not code for anything or does not code for protein? 2%, 25%, 50%, or 98%? Well, it wouldn't be very interesting if it was the first three, but it's the last one. And so that's something I think is pretty interesting, that 98% of your DNA is non-genetic. 98% of every chromosome in every cell of your body doesn't code for protein. That's crazy. Now, we'll say this. Does that 98%, is it just wasted? No, it has other structural functions, and it helps to protect the ends of the chromosome and things like that. But also some of it is just a legacy from our evolutionary past. So anyway, that's pretty interesting that you've got all these cells with all this DNA, most of which doesn't code for anything. That's pretty cool. Okay, let's talk about transcription. So the first step is to actually find a gene. And so in humans, we've got 25,000 genes, but like we just said, most of our chromosomes are non-genetic. So the first step is to simply find a piece of chromosome that actually will code for RNA and then protein. So finding a gene is sort of the first step. Is most of your DNA genes? No, it's not. We just said that. Most of your DNA does not code for anything. And so how do we know where the gene is? Most genes, all genes, can be recognized by a unique starting sequence. And so the sequence of DNA is always important, but to know where the gene starts, it has a unique starting sequence which is called a promoter. And there are several of these, and if you take genetics, you get into a lot more detail about these. But that's how you find the start of a gene. And there's an enzyme called RNA polymerase which finds the promoter and then starts transcribing the gene because that promoter marks where the start of the gene is. Now you may remember that we talked about DNA polymerase earlier, right? You remember DNA polymerase read one strand of DNA and filled in the other strand. RNA polymerase does the same thing but with RNA. It reads one strand of DNA and it fills in a piece of RNA based upon that strand. And just like with DNA, remember that if we knew one strand of DNA, we automatically knew the other because A's always paired with T's and C's always paired with G's. All tigers can growl. Well, the same thing works for RNA, right? We can read one strand of DNA and build a piece of RNA based upon that same concept. So that's basically how it works. That's how DNA tells us what piece of RNA we need, is because the sequence of DNA can be used to build that RNA because we know how the base pairs pair up. And so again, RNA and DNA are both nucleic acids. They're very similar. There are a couple of important differences though. The first is RNA is single-stranded. Remember that DNA had two strands, right? And it had the two strands and then they made the double helix. RNA is only a single strand. Another very important difference is RNA uses A, C, and G, just like DNA, but it uses uracil, a U instead of T, instead of thymine. So it's got a slightly different nitrogenous base. Now, those bases in RNA are a little bit different. They use a different sugar. They use ribonucleic, or ribose as their sugar. DNA uses deoxyribose. But the important thing to know is that when you look at those nucleotides, when you look at those bases, in RNA you've got A, C, G, and U. And the U pairs with the A. And the C still pairs with the G. Okay, let's look at an example. Here's just a figure from your book kind of showing you DNA's got two strands and the nucleotides are pairing up, C's with G's, A's with T's. RNA is a single strand. And if you look at this, the code of this RNA in this example, you see you've got C's and you've got G's and you've got A's, but you don't have any T's. Instead of T's, you got U's. Okay, so let's practice. Let's just look. So this is a really short piece of a gene. Most of human genes have 20 or 30,000 base pairs. Some have 2 million base pairs, right? And here we've got 6. But obviously, we don't need many to do an example here. So this is a piece of DNA from the gene. And you see it's two-stranded, and you see the A's are with the T's and the C's are with the G's, just like we expect. So RNA polymerase will find that promoter and will read only one of these strands. So only one of these strands is actually used to make a protein. The other one is just there. And so in this example, the RNA polymerase is reading the right-hand strand in this particular example. And you see how it does just like DNA polymerase did. It reads the strand. Whatever nucleotide it reads in the DNA tells it which nucleotide to bring in the RNA. So the first thing it reads is an A. A's pair with T's. Oh, wait a minute. RNAs don't have T's, they have U's. So the RNA polymerase brings in a U. Reads the next one. It's a T. Oh, T's are with A's. I can bring in an A. Make part of my RNA strand. G with C, C with G, G with C, A with U again. And so you see the green boxes here, that's the new piece of RNA we're building. And that's it, that's all we need, because it's only a single strand. And again, why is that a U instead of a T? Because RNA has a slightly different base there. It uses uracil, not thymine. But it's still that U pairs up with the A. So that's just a little bit different between DNA and RNA. And so there's the piece of RNA that we got from this gene. Again, this very, very short piece of a gene. And so we can take that RNA and go make a protein with it, translate it to a protein. And so here's a figure from your book, again, kind of showing you this general idea. You see that RNA polymerase is going along the DNA strand. It kind of pulls it apart, and it's reading one strand, and it's bringing in RNA nucleotides, and it's building a piece of RNA. And so in real life, there's a lot of steps, and it's really complicated, but that, I really want you to just understand the basics here, right? For a non-biologist, it's important to just understand the basic idea of how DNA makes RNA. So the RNA polymerase, RNA poly, finds the promoter, which tells it it's the start of the gene. Then it reads one strand of the gene, and it builds a new RNA molecule based upon the strand it's reading. Now, one of the things that we also want to talk about is that some molecules bind to that promoter in such a way that the RNA polymerase cannot bind to the promoter, right? So think about that. So you've got this gene, and there's this promoter that tells, this is the start of the gene, and that's what RNA polymerase has to find to get started. But here's a different molecule, a different enzyme, or some kind of different protein. Or it doesn't have to be a protein, it could be a lot of different molecules do this. And it binds to that promoter. Well, if it's bound to that promoter, then RNA polymerase can't bind to the promoter. If RNA polymerase can't bind to the promoter, then the gene can't be transcribed. If the gene can't be transcribed, it can't be translated into protein. That gene is effectively turned off. And you say, big deal. Well, it is a big deal, because this is really important to how your cells function, right? So think about this. What's a big deal? What's something that I mentioned earlier when we talked about cells? Every one of your cells has all your DNA in it, right? So a skin cell, an intestine cell, a muscle cell, a big toe cell, I don't know if there's big toe cells, they all have all your DNA, right? All 46 chromosomes. And yet, they all do different things, right? One turns into skin, and one turns into intestine, and one turns into a big toe, and whatever. Okay? Well, how? It's because certain genes get turned on, and other genes get turned off, and that determines what the cell does. And so, if there were big toe genes, they get turned on, and the cell becomes a big toe. And that's crazy, there's not a big toe, but don't go telling everybody that there's big toe genes. But I'm just making a point. Anyway, genetic control is incredibly important. Like what genes get turned on, what genes get turned off, when they get turned on, when do they get turned off. That is very important to determining what your cells do. So this idea that you can turn a gene on and off is really important. And what do I mean by that? What do I mean by a gene is turned off? Well, what's a gene's only job? To make RNA, which will make protein. If the gene can't make RNA and can't make protein, then the gene is off. If the gene can make RNA and can make protein, then the gene is on. Well, how do you turn a gene on and off? One way, an important way, is to control it at this promoter. So this is a pretty fantastic idea. Now, in a similar manner, there are molecules that are promoters that can help RNA polymerase bind to the promoter. So the promoter is the sequence at the start of the gene, but they're molecules that help promote the promoter. I don't want to confuse you there. Some molecules can bind to the promoter and help RNA polymerase, and so can enhance genetic production, and so can help turn the gene on. And so that's going on in all of your cells, right? And so here's a figure from your book kind of showing you that idea. At the top, you've got repression of transcription. You've got something binding to the promoter, blocking RNA polymerase. The gene can't be transcribed. At the bottom, you've got activation of transcription. You've got an activator. I guess activator, I said promoter earlier. Activator is a better word here. An activator which binds to the promoter, which helps RNA polymerase bind, which turns that gene on. So we'll see in examples throughout the rest of the semester how this is, pretty important. Okay. So that is taking a piece of DNA and turning it into RNA. That was transcription. Now let's translate that piece of RNA. And so now we're going from one language to another language. We're going from a nucleic acid to a protein, two very different molecules. Well, you'll recall that the building block of a protein is an amino acid. And so we put a bunch of amino acids together in a line, they all hold hands, and then that long string of amino acids will kind of curl up into a three-dimensional shape, and then you've got a protein. There's only 20 of those amino acids that get used. So just like there's 26 letters in the alphabet, in the English alphabet, and you can make any word from those 26 letters, there's only 20 amino acids. You can make any protein you need from these 20 amino acids. You string them together, you get a long string of them, that's a protein. But how do we know what amino acids to use and in what order they go, right? We can't just string them together at random. The particular amino acids you use and the order that they go in is critical. Just like if you're writing, writing some words down, right? Which letters you use and the order of the letters is critical to whatever you're writing. It's the same thing here. Which amino acids you use and what order they go in is critical to make it a protein. So how do we know which ones to use? Well, the RNA tells us. Well, how does the RNA know what to use? Well, the RNA just got its information from the DNA, and that's how DNA controls everything in the cell. Okay, well, how does the RNA use its information to make this protein? Again, there's lots and lots of details here, but we don't need to get into all those details. We just need the 10,000-foot view. We need the big picture here. There's an organelle called a ribosome. So, organelles, little structures inside every cell. And the ribosome can read the RNA. Remember how we had like DNA polymerase can read DNA. RNA polymerase can read RNA. Ribosome can read RNA. But it reads it a little bit differently. So every cell has ribosomes. The ribosomes read that RNA and use that information to build a protein. I mean, it's just like we did before, where we had RNA polymerase read the DNA, used that information to build an RNA molecule. The ribosome reads the RNA molecule, uses that information to build a protein. But like I said, it reads it a little differently. Whereas RNA polymerase went base by base and matched up every base, right? Because we were transcribing. We were just going from one nucleic acid to another. So it just went base by base. Now we're translating. Now we have to take the bases and read them a little differently and put them into a different type of molecule. And so we don't read them base by base. We read them 3 bases at a time. And so it's like, again, it's like, to use the analogy of a language or words, it's like every word is only 3 letters long, and they all mean something, and they all mean a particular amino acid. And so the ribosome reads the RNA 3 bases at a time, and that tells the ribosome which amino acid is needed. And those 3 bases at a time is called a codon. And, excuse me, so here's the piece of RNA that we built earlier. Instead of going letter by letter, we take 3 bases at a time, and that's a codon, UAC. That tells us what amino acid goes there. Excuse me. And then the next 3 base pair is also a codon, GCU. That's a different amino acid. And the ribosome is able to read these codons and know what amino acid comes in and build the protein in that way. So each codon tells us what amino acid to add to that growing protein. And so in this particular example, the ribosome reads UAC and it goes, oh, tyrosine. So it brings in a tyrosine. Then it reads GCU, oh, alanine, and it brings in an alanine. And it binds the alanine to the tyrosine. And then it just brings in the next amino acid and the next one. And it makes a long chain of amino acids. Now, something I don't know if I've emphasized yet, but we have talked about when we talked about these molecules. Think about the structure of these molecules. Remember we talked about the structure of DNA, that double helix, but it's two long, straight molecules that are, two long, straight strands that are bound together to make one molecule of DNA. And then RNA is one straight strand of nucleic acid. And you can see why those two are very similar, right? Because you're taking one strand of DNA and you're using it to make one strand of RNA. But now you're taking one long straight strand of RNA and you're making one long straight strand of amino acids, which is going to be your protein. They're all very similar because they all, rely on one another. One builds the next, builds the next. That's why they're not branched. They're not, when we talked about carbohydrates, you remember how we said sometimes they form like these rings and stuff like that? No, these things are all just straight, long strings of different bases holding hands together. And then you end up with amino acids holding hands together in a long, straight string. But then that string, as far as the protein's concerned, then that string can wrap up and turn around and make a crazy three-dimensional shape and make it an active protein. So when you, again, why do we make a big deal about the structure? Because once you understand the structure, you see how all this stuff is related. Okay, anyway. So the ribosome reads that RNA 3 bases at a time. It tells the ribosome which amino acid to attach, and then a long string of amino acids, there's your protein. And then what do we say the protein does? Do you remember in the last lecture, proteins do a lot of things. They store energy, or they make muscles, or they make collagen in your skin. And very often they make enzymes, right? And that protein is an enzyme that goes and helps a chemical reaction, some chemical reaction in your cells. And so that was 6 bases in a gene, an example. And that made a 6-base piece of RNA. And that made a protein that only had 2 amino acids, right? Because you have to read the RNA 3 bases at a time for 1 amino acid. In real life, of course, the gene is thousands of base pairs, and the RNA is thousands of base pairs, and it makes thousands of codons, it makes a string of thousands of amino acids, right? These are big, long, complicated molecules, but it's the same process, just, a lot longer. And that's it. And there's this big mystery, this big important idea in biology, DNA makes RNA makes protein. We've basically covered it, right? Are there a lot of details? Yeah, there are a lot of details. But for our purposes, this is the most important thing here. Now, we'll get into some details later, a little bit more details. Now you can sort of see, we talked about mutation, right? And we talked about mutation as a change in the DNA. There's lots of ways that mutation could happen. But now you can put this all together in your brain, right? If that DNA changes, if a base in that DNA changes, well, that's going to change the RNA, right? Because of transcription. And if that RNA changes, well, that's going to change the codon, and that might change the amino acid. And if you change the amino acid, you change the protein. So you can see how those mutations can lead to new proteins potentially, and that's hugely important to biology. And so again, we're just going to, just to reinforce these ideas. Here's a piece of RNA. How can we translate this into a polypeptide? We've got to read it by codons, 3 base pairs at a time. We're taking one language and converting it to a different language. So we're taking it from the nucleic acid language and converting it to the protein language. That's translation. We need to take words, quote unquote, from the RNA and translate them into amino acids for a polypeptide. Those words are 3-letter groups called codons. And so the first codon in this example codes for tryptophan. The next one codes for phenylalanine, the next one for glycine, the next one for serine, and so on and so on and so on. Now, how do I know this? How do I look at this and I go, oh, that's a phenylalanine? How does the ribosome know that? How does anybody know that? The interesting thing is, codons always code for a particular amino acid. So for example, GGC is always glycine, no matter what the organism, no matter where you are. Now that's cool, and that has a deep, profound meaning that we're going to get into. Now, let me say that again, because it's worth repeating. The codons always code for the same amino acids, no matter what the species. And so once scientists figured out those codons, now you can take any piece of DNA. Of course, you can easily figure out the piece of RNA, and now from that RNA, you know the sequence of amino acids, because you always know what that codon means. And that's how the ribosome always knows. You know, and the ribosome doesn't know, right? The ribosome doesn't have knowledge. The ribosome is just helping along a chemical reaction. And so here is that code. And this is what's known as the universal genetic code. And so if you look at this, this is taking all possible codons from a piece of RNA and telling you what amino acid they code for, right? So we figured this out a long time ago. And so if you start on the left, you got the first base, the top is the second base, the right is the third base. At any rate, for any three-letter combination of RNA, you can use this chart to figure out what is the proper amino acid. And that's pretty cool. Now, if you look here, there's 64 possible codons, but only 20 amino acids. And so how do I know there's 64 possible codons? Well, there's only 4 bases in RNA. And to get a codon, you're taking them 3 at a time. So if you do the math on that, there's 64 combinations. And if you look at this chart, if you count up every unique codon in this chart, you'll see there's 64 of them. But there's only 20 amino acids. What's that mean? That means that each amino acid has multiple codons. And so pick any amino acid in this chart, and you'll see that, oh, there's several different codons that will give me the same amino acid. There's a couple of start codons, a couple of stop codons. So the ribosome knows where to start making the protein, where to stop making the protein. Now, if you chew on this a little bit, you might understand something that I've been saying all semester. So when I talk about a mutation, I say a mutation is a change in the DNA, and that will change the RNA. But then I always say it might change the protein. Well, this is why it might change the protein. This is one reason. Because you might mutate the DNA, which will definitely change the RNA, but it's possible that the new codon you created still codes for the exact same amino acid. And so you've mutated the DNA, but you still get the same amino acid in the same place. And so that's what we call a silent mutation. So even though the DNA changed, it didn't change the protein. Now there's other things that can happen too, but now you're starting to understand why I say it might change the protein. Now remember, the really interesting thing is this code is the same for every living organism. And that is deeply profound and very cool. So, if a ribosome in one of your cells is translating and making a protein, and it reads AAA, for example, so it's got a piece of RNA, it reads AAA, it says, oh, lysine goes in here, right? So that's in your cell. But if you had a cactus, and it had a ribosome, and it was making a protein, and it read AAA in that codon, then it would also stick in a lysine. Right? You've got two very different organisms, but they use the exact same genetic code. And that is really cool. Okay, so again, the ribosome is looking for that start codon, and that tells it where to begin the polypeptide, right? So just like the RNA polymerase was looking for a promoter, the ribosome is looking for a start codon. Then it reads the codons one at a time, and it builds the polypeptide by adding the proper amino acids based upon the codon. And how does it know what's the proper amino acid? Because that universal genetic code. No matter what organism, no matter where you're at, it's always going to put in the same amino acid based upon a particular codon. And then it keeps going until it hits a stop codon. When it hits a stop codon, boom, now you've got a string of amino acids, now you've got a protein. And so you see how we're translating the RNA into a new language. We're taking the nucleic acid and we're putting it and building a protein from it. And you see how we're building a string of amino acids based upon the information in the RNA. And where did that RNA get its information from? It got it from the DNA. So now you see how the DNA is able to run the show. And so here's just another schematic of a ribosome, just kind of showing you. It's this organelle. It's made of protein and RNA, and it of itself is made of RNA. And you see that there's a piece of RNA in the ribosome, and it's reading it 3 at a time, and it's bringing in amino acids and having a growing polypeptide. And so here again, just to put it all together, you've got this gene, and you zoom in on the gene, you can see the DNA double strand, which gets transcribed into RNA, which gets translated into a string of amino acids, which is a protein. And so that's it. You know, there's lots of details here, but you have the basic idea. You know RNA to protein. The ribosome just reads the codons on the RNA and uses that information to add the proper amino acid to the growing polypeptide. Then it shifts to the next codon and gets the next amino acid and does that till it hits a stop codon. And so we've talked about all the steps here now. You've talked about DNA to RNA to protein. You can call yourself a biologist because you've got the basic understanding of the most important idea in biology. All right, last thing I want to talk about is let's talk a little more about this universal genetic code. Okay, so why do I keep making such a big deal about the universal genetic code? Well, first off, I just think it's interesting. All living things function in this way. And that's just pretty cool. When you think about the diversity of life that's existed on this planet, the diversity that exists now, from a bacterium to a human, to a pine tree, to a jellyfish, all living things function this way. And that's pretty cool. Now, do you mean some do and some don't? You know, am I exaggerating? No. All living things. Okay. You never say all things in biology. There's always something in biology that's going to test your statement. And there's a very few examples where a codon like codes for a stop codon instead of an amino acid in a couple of organisms. Very, very rare. But if you look at all the species and all the DNA and all the genes, yeah, everybody does the same genetic code. And that's pretty cool. Now, while gene expression differs among all the living things on the planet, this idea of a gene is universal. And so that's what I'm trying to say here. This idea of taking a piece of DNA, making RNA, making protein, is shared by every species. And that is just one of those really cool things about the world when you learn about it. But it also has like some kind of cool practical applications. For example, you can take a gene from one species and put it into a different species and it'll work. It'll make the same protein because of this universal genetic code. And that's pretty cool. We'll talk about that in a little bit more detail. And the reason that this works is because of this thing that's shared by all living things. You can splice a piece of a gene from a chromosome. You can take a gene from an organism, splice it into a chromosome of a different organism, a different species, and it will make RNA and then make protein in the new species. Now, not automatically, right? There has to be conditions that are met, but if you can meet those conditions, the machinery and the code is exactly the same. Because DNA to RNA to protein works the same in every living thing. Now, here's another really cool thing. Like, what does this suggest? All living things have their very basic biological mechanism is exactly the same. That suggests that all living things have a common ancestor. How do we get shared traits? We inherit them from our earlier ancestors. And even though you and a bacterium are very, very different, you still have this one thing that you share with that bacterium. What does that suggest? If you trace your lineages all the way back, somewhere a few billion years ago, you came from the same common early ancestor. And that's really cool. And there's some practical applications of this again. Like I said, for example, we talked about viruses. Now, viruses, we say, aren't living things, but they have a lot of shared characteristics with living things. But how can a virus get into your cells and make its own proteins? Because we all make proteins in the same way. And so even this lowly virus is not really a living thing. It can get into your cells, it can get into your DNA, and it can make its own proteins because of the shared genetic code. But a really cool thing is what we talked about a few slides ago, is transgenic organisms. And so again, you could take a gene from one organism, put it into a different organism, and make it work. Now, it's not that simple, but then again, it is kind of that simple, right? If you put it into the right chromosome with the right promoter under the right conditions, it will make the same protein in the new species as it does in the original species. And that's a transgenic organism. The term that you're probably familiar with is GMO, genetically modified organisms. And we're going to talk about these quite a bit. But the basic idea is we're taking a gene from one species and putting it into another species. Why does this work? Because all species share the same genetic code. And so you can get cool stuff like this. And so here's a tobacco plant that has a firefly gene in it, right? So fireflies have a gene that makes an enzyme called luciferase, and that enzyme causes a chemical reaction that ultimately can cause a light to fluoresce, and that's how lightning bugs can light up. Well, you can take that same gene, that luciferase gene from a firefly, splice it into a tobacco plant, and the tobacco plant will light up because that gene gets transcribed and translated the same way in the tobacco plant as it did in the firefly. That's pretty cool. Or here you got a pig. If you look at this pig, look at its snout and its feet, its hooves. You've got a jellyfish gene. You know, again, lots of organisms are bioluminescent, can create, can luminesce, or maybe this is just a pigment. I'm not really sure about the jellyfish gene. But again, this is a gene from a jellyfish that got spliced into the chromosomes of this pig, and the pig is making the same gene product as the jellyfish and expressing it in their nose and in their hooves. That's really cool. And so we'll talk about GMOs, transgenic organisms, in a little bit more detail upcoming, but this is why they work, because the genetic code is universal to all living things and to all species. And that's a really profound idea. So anyway, okay, that's the basic idea behind transcription and translation, and we're going to build upon that idea a little bit. But as always, if you've got any questions, please let me know. Otherwise, have a good day, and I'll see you later.